In this video, we are going to discuss about nature-inspired computing technique subject, and I'm Deepa from SRM University. Here we are going to discuss about immunocomputing, and in immunocomputing, we are going to focus on pattern recognition and binding. So we already discussed what is an immune system and what are the components of immune system, right? An immune system is one which uh, act against the pathogens. So whatever the disease causing agent, it might be either bacteria or virus or something, some pathogen, if it enters into a body, our body has an inbuilt structure that will act against this pathogens. Okay, so that is called immune system. Now we'll see how it is actually doing. Okay, so the first step in this fighting against the pathogen is pattern recognition and binding. Okay, so pattern recognition in the sense like it is going to recognize some foreign agent entered into a body, and what is the structure, molecular pattern of that entered pathogen. So that need to be detected. So that process of detection is called pattern recognition and binding. So here, like when some bacteria, some a pathogen enter into a body, some portion of those pathogens are recognized by the immune system. So that portion of those pathogen, right, that is called an antigen. Okay, so this antigen or a type of pathogens or a set of molecules of pathogen that are recognized by the immune system. Okay, so here what happened now, like this antigens, whatever the bacteria or virus, it has a receptor molecule in its cell. Okay, so that surface that receptors, we call it as an epitopes. So here our B cell will attach with those epitopes. So this is one example structure. See, this antigen or a pathogen, it has some set of receptors. Okay, so that presents in the surface of the antigen, like either a bacterial cell it might be any kind of pathogen. So it has certain type of receptors. Okay, we call that as an epitopes. So our blood cells, okay, our immune cells, it might be either a B cell or a T cell. It has a receptor for that. Okay, and remember antigens, it has a variety of patterns for this receptor, whereas your B cell or a B cell receptor, it is monospecific. Okay, so all the receptors for a B cell, it is of the same structure, right? Okay, when it is an arrow mark, it is all arrow mark. When it is a U shape, everything is of U shape. Okay, so here this receptor will attach with the epitopes of an antigen. Okay, so once it is attached, that recognition between that receptor and the epitopes, right, we call that as an AT. Okay, so antigen, you have various patterns of epitopes, whereas in a basal, it is the receptors are of monospecific. All the structures are of same type. And here, this B cell receptor, we call it as an either antibody or a immunoglobulin. Okay, this B cell receptor, we call it as an either antibody or a immunoglobulin. And this receptor, it is actually formed by a different combination of genes. Okay, so this is one example. Okay, these gene libraries will be there existing in the immune system. Okay, so whenever a pathogen occurs, whenever an attack occurs, the receptors are formed by different combination of this gene library. Okay, so that is automatically formed, and one might matches with the epitopes. So whichever matches, that receptor is going to recognize the antigens more pattern. So here the attraction between the B cell receptor and epitopes of antigen, so that is called affinity. Okay, so whenever you have a better recognition, you have the higher affinity. The interaction is more. So when the interaction is more, the receptor has the capability of getting the entire molecular pattern of the antigen present. Okay, so this affinity is an important concept that is there in this artificial immune system concept. So here, when the receptors are covered, okay, so just recognizing is not a matter, right? Once we recognize some pathogens occur, okay, we need to destroy those cells. So the destruction is done by so much of antibody that actually clubs together. Okay, the nearby antibody will be clubbed together and it will cover up the antigen, okay, cover up the pathogen, and it will destroy the pathogen cell. Okay, so this is how a pattern recognition binding occurs in terms of an immune system. Okay, thank you.